So I'm working at Karolinska Institute. I'm associate professor in stem cell biology. And I would like to talk to you about where we are in the research front in genomic science today, and what can be, or what is close to actually translate into the real treatment. So genomic science have been going on for a while, but it's quite recent it reached humans. So here we have some cloned frogs from the 1950s, 60s. And the cloning is a process where you actually take the genome from one individual or organism, and then you clone that. So you get a clone of with the same DNA. And frogs were cloned then in the 50s, 60s. We have the chip dolly here in the 90s. And then it have followed cloning of animals and for different reasons. So cloning horses would be done because for many reasons, I would say. So a very good racehorse, of course, is it's worth a lot of money. And if you can clone that one, you can have several clones and actually sell them. Then there have been cloning of animals, or domestic animals or pets. Here we have the rainbow that have been cloned, and the copycat. So if you have a lot of money, maybe you want to have your pet back again. And as you can see here, the problem is that it's not only the genome that is responsible for how you look and so on. And here the copycat doesn't look exactly as its mother, or the clone doesn't look as similar. And this is because of environment and epigenetics, which I will not discuss here. And then, of course, you can clone extinguished animals, which have been done. And this is a way to keep them when they are almost extinguished from the planet. But it wasn't until 2018, the same process of cloning, somatic cell nucleus transfer, was done in the human setting. And the idea here is that you have a skin cell and you take out the nucleus or the whole cell and put it into a fertilized egg where you have removed all the genetic material first. So the genetic material from this skin cell of an adult individual would be coming into the environment of a fertilized egg. And of course that environment is programmed to create an embryo and further then a new individual. However, of course, in humans, this is not this stage, which is an embryo in vitro in the cell culture disk, can't be implanted to a pregnant woman to continue the development, but these cells here can be taken out and cultured. And these cells then are clones of cells from the individual that gave the skin cells, and these embryonic stem cells can be a lot of different cells in the body. Okay, so if we're not allowed to clone humans or babies, what are we then going to use this technique for? And the idea is that, these type of technique have the potential to cure incurable diseases. And here is a lot of buzzwords that is used nowadays, and it's precision diagnosis, personal medicine, replacing injured cells, dying cells, revert and heal dying cells or sick cells, to correct mutation that cause disease, or to modify our immune system so it kills cancer. And all of these new ways of looking at medicine. So all these are novel approaches to medicine and health. And it includes then a novel view of medicine and health. And I will go more into that. So if you think of disease, might before have been thought of as linear. So either you're healthy, then you become diseased, and that is and the disease will be not really cured, but actually treated to minimize symptoms. for some diseases you're cured. Let's say you get a bacterial infection and you take antibiotics and then you're cured. But for many cases you might not. And those diseases are called incurable then. So, but if the idea of health and disease would be circular, then you're either in the healthy part and then you go over to risk part. And at this point, you are in risk of a disease. And then you get disease and maybe you can cure it and you're back into the healthy. And genomics and so gene and cell therapy comes into this circle at different places. So when it goes to the risk, when the person is in the risk part, the risk can, in many cases now, it seems like the risk of getting a disease, sequencing of the genome might be able to pinpoint the risk individuals in the risk part of this circle. And then, of course, precision medicine, personal medicine. So depending on what risk and what disease you're at risk of, you will get a treatment that is kind of dedicated for you or for a couple of, a group of patients with the same similar type of risk for a disease. And in some cases, it might be possible to actually remove the gene that you find the gene already here in a person in risk of getting Parkinson's disease. But of course, when you get the symptom of Parkinson's disease, already 80–90% of the cells that produce dopamine have died. So if you can go in early, if there is a genetic cause, and then actually do a gene therapy treatment. And let's say you come here to the disease, you have type 1 diabetes and all your insulin producing cells are gone, then maybe a cell therapy would actually be something that can cure you. You get new dopamine or insulin producing cells at the point, and then you don't need treatment, but you actually go on to the health part here. And then there is another part, upcoming treatment in the same area, and it's the immunotherapy for cancer, which is both cell and gene therapy. So just say these parts with the risk and the disease part of the circle might be easier to discuss regulation and ethics because it's an easier view of what is needed and why it's needed and why it's important. And maybe this part might be more tricky, the healthy individuals and how to apply genomic research there, because a lot of the sequencing type of research, the idea with that is to learn how can we find the risk people, and maybe also then healthy people would be needed to sequence and so on for this. So what have what have led us, so now I will go back a little bit to what have led us in to this novel view of medicine, actually, that you can maybe treat and cure diseases. And there are several, of course, findings, breakthrough findings, but in this talk I will focus on two different findings, and those findings have been awarded with the Nobel Prize. It was 2012 that John Gurdon and Shinya Yamanaka got the Nobel Prize for the discovery that a mature cell can be reprogrammed to become pluripotent. And that finding have been key findings in the cell therapy or cell replacement therapy route. And then we had the 2020 Nobel Prize by Professors Emanuela Carpenter and Jennifer Doudna about, or the prize for the development of a method for genome editing, and in this case, the CRISPR-Cas9 technique. Okay, so first I will go into the 2012 Nobel Prize that opened up for cell replacement therapies. So, and here we're back a little bit to the cloning, because of course John Gurdon did the cloning in the 60s, and then Shinya Yamanaka did the IPS reprogram in 2006, but both got the prize at the same time. And the idea is that development is a one-way road, where you have the stem cell up on a hill, and it's rolling down, and then it's making route choices to go down this way or that way. And once the cell have done this, it can never go back. So this cell can only be this cell type or that cell type and not go down here. And once you're down here, you're this special cell type. And this is Waddington suggested this 1956. So it's called Waddington's landscape. And similarly, up here on the hill, it's the fertilized egg, and the development is one-way route, and you develop a human with many special cells. And here are the special cells. And also, so how about the genome? So the interesting thing here was that before, at the 1956, around there, or earlier, maybe 1940, 38, I think was formulated. The idea is that when the cell is specializing, what is happening to the genes in the genome? And 1940, then scientists thought, like, to become a liver cell, let's see here, you have a liver cell, you have to remove genes that is important for creating muscle, nerve, skin, for example. So they are off. Or at that point, you thought that those genes were thrown out of the cells. The stem cell up here has everything open, genes, and can become everything. So the question is the genes here for the other cell types, once you're down here specialized, are they thrown out from the cells or not? Actually not. So now we have a completely new way, after especially the cloning have shown that all the genes are there, because the skin cell can actually create a clone of another organism or a clone of itself, but also with the reprogram. So this is how it's viewed now. The cells are not statically down here as a skin cell. It can actually be pushed back this mountain and go up here and actually then choose another route down, etc. So this is, and the interesting thing is then, of course, and this is what happened on the gene level. The genes were off. So we have a liver cell down here. All the genes for not liver cells, like muscle, nerve, and so on, is off. But you can push the cell back and actually switch on all these genes needed for this cell to be able to open up for any fate. So this fountain of youth is not really science fiction, because what's happening, which I think is even more interesting, I will not discuss too much here. When you push a cell backward in development like this, you are also humanizing it. So this cell is becoming young. And that's, of course, interest. So scientists can humanize cells on the cell level, not really whole organisms yet. So reprogramming then to guide development backwards. So how is this done? How do you push a cell from down here up here? So just to show you again what type of cell we are talking about. So we have a fertilized egg, we have the human embryo, 5 days old. This little clump of cells is taken out and you grow embryonic stem cells. You can grow them forever. This was done first time 1998 in humans. However, there is another way to produce or create this type of cell, which is not coming from the embryo. And of course, these ES cells, then embryonic stem cells, they can grow forever and they can create all cells in the body. But you can create them like this. You take a skin biopsy from an individual, and this person can be, of course, a donor with a disease or a healthy donor. So if it has a disease and the genome is causing the disease, that genome will continue being present in the cells. In the skin cells, you reprogram the cells where you push the cell up this hill, and that you do by introducing some genes, which are important for opening up all these switches. All the switches that are to off, the gene is off, need to be on, switched on. And there are some potent genes that Yamanaka discovered that do this. Then you have stem cells. Similar stem cells, they are called induced pluripotent stem cells because they are induced and not coming from the embryo. These can be grown also forever. They are immortal. You can freeze them down and keep them in the liquid nitrogen for 10, 20, 30 years. You can specialize them. So you can get as much neuron as you want from these cells. So from this individual, we can produce as many neural cells as we want, or as many cardio cells as we want. Yeah, and these are the definition: expand, definitive, and become any cell of the body. Now I go into the next Nobel Prize, and after this I will connect these two together to discuss what we can do with this, both the technique. So genome editing and cutting and pasting in the DNA, correcting, removing, or introducing genetic material in the cell. You have a cell, you have the chromosome, and you have a technique where you put in a guide molecule that finds the right place, the mutated gene in the genome. There is an enzyme coming with it. It cuts out this mutated gene and corrects the DNA. And this is really precision, doing this with a precision. All techniques before the CRISPR-Cas9 was quite blunt, and you never really know exactly what you got. Okay, so we have techniques to cure incurable disease, the techniques of reprogramming, genome editing, sequencing, because the sequencing is important, as I said, to find who will we treat, maybe already in the risk period before presenting disease. Have the potential to revolutionize medicine, so precision diagnosis, precision and personalized medicine, novel treatment that never been available before, cell replacement therapies, gene therapies, immunotherapies. And some of these have started to get into first in man trials and so on, but still there is not routinely used. So cell replacement therapy for different diseases, you have seen this before. And in this case, we produce a lot of iPS cells. We take the skin biopsy and reprogram, and then we can create cardio cells, neural cells, retina cells for eye treatment, for Parkinson treatment, for cardiomyoinfarction treatment, for example. And this should be done at the hospital, and the idea is to build up a bank of cells from different donor individuals, because of course it's similar here. If you're going to treat a person with a cell therapy, that needs to be matching to the patient, similarly as blood banking and matching with the AB system. Also here it has to be matching. So there need to be several of these individuals to build up bank of the iPS cells. Then of course you can do same iPS cells, you can build up a bank of dopamine neurons to be shipped to the hospital for the treatment. And similar, these cells can also be used for making cardio cells and shipped for treatment. And of course this whole process is complicated and new and so on. And there is a lot of steps involved that it doesn't have to do too much with basic science anymore. Treating disease with gene therapy. Here is an example of a disease in lung, maybe cystic fibrosis. You put the CRISPR-Cas system into a viral vector, and this virus is non-harmless virus. The only task for the virus is to go via an inhalation nose spray into the lung, enter the lung cells, and provide the CRISPR system to the cells, so the CRISPR system can cut out a gene and correct a gene mutation. Problem here is then this treating, doing the genome editing in cells that will affect future offspring is of course more come with more challenges than treating and correcting genes in some only one cell type. So let me see here. And then we have the immune therapy, which is a really elegant way to treat cancer with gene therapy and cell therapy. So what is done here is that T cells, which is the killer cell of the immune system that kills other cells, but not really cancer cells, because they don't kill cancer cells in normal case because they are your own cells. But what you do, you take out T cells, you introduce with CRISPR-Cas9 technique a receptor that is called CAR, and this receptor sits then on the T cell, and this receptor is specific for the cancer that the individual has. And now the T cell is programmed to actually attach to these cancer cells and kill them. And these cells need to be then expanded to grow up in the cell lab, and then introduced back into the patient, where the T cells that have been modified is then killing off the cancer, and then these cells the T cells will not stay too long because you get new blood cells all the time. So these cells will just go in and do their job, and then after a couple of weeks they are gone from the patient. So patient-specific combination of cell therapy and gene therapy, it's also discussing in the way, for example, neurodegenerative disorder like Parkinson. You take a skin cell from the patient, you reprogram to iPS cells, you use the CRISPR-Cas9 technique if you need to correct a mutation that maybe caused this Parkinson, and then you grow up these cells, you produce a large amount of nerve cells, and then you transplant back to the patient. But is this the future, really? Maybe not. This process is expensive, resource, labor intensive, and resource costs a lot. So there are different things that are discussed. Is, of course, you do donate cells to yourself for cell replacement therapy, as I just showed, or we have donors that donate to several, where you can have a bank of iPS cells in frozen liquid nitrogen tanks, where you take up one dose of cell therapy that is sent to the hospital for treatment. And the hospital doesn't need to be expert on all the technique involved. But of course, it's a logistic and quite complicated process. And I would like to show you this in the end here a bit about, like, okay, so how do we translate? And this is actually, today, this is true. So if you look at this one, we are curing animals with diabetes, Parkinson's disorder, macular degeneration in eye, and so on. So basic scientists are treating animals and curing animals from these disorders. How do we get over to curing humans? I mean, using similar way as curing the animals. And there is actually a huge translational gap here, where we as basic scientists, we kind of look at our treatments and our research going down in between here. It never really bridged over here. And this is a big frustration actually among all scientists. And one of the biggest problem is the language is not really the same. And the hurdles here, because the people standing on this side that should receive this, they don't have everything in place when it comes to regulation and techniques and competence to actually bridge it over. And it's a lot of money. This is a money gap too. There is grants for basic science, but nothing for translation really. So curing animals, curing humans, and building bridges, it's kind of a lot that needs to be done, and there is not too many bridges built yet. And of course, there is this thing I work a bit with this in different settings. So and it's really a communication cooperation type of project to bridge this gap. and resources if needed. And when I say communication and cooperation, it's because there is so many stakeholders involved that need to talk to each other and understand. The translation scientists need to understand what the basic scientists need to bridge from their side, so they can bridge from the other side and meet. And this involves, of course, ethical and regulatory challenges, much of that. Yeah, so let me see if so then I would like to thank you for listening. Here I have an acknowledgement of the research group that I'm heading, but also the IPS core facility, which I'm directing also. And thank you for listening.